Yeah, well, Annemieke, that's very early.
I know, but I'm giving lectures all day. So one lecture just finished. So I thought I'm just gonna log in and I can email while I wait.
Is running. I've just shared the YouTube
Okay, very
address
good.
on Twitter. And yeah, so
Yep.
we're up and running.
Great.
So I think things will be okay. So I suppose that you see the holding slides now instead
Yeah,
of me?
I see the
Yeah.
holding slide.
Okay, that's all right.
Yeah.
Okay, so do you want to try and share your slide before we start?
Oh, we
I
can
can
do this.
stop my share and you can start your
When you
slideshow.
share, I cannot share.
Let me do something more. Make host cancel.
You have to stop sharing first.
No, I've done it.
But yeah.
But you're now the co-host. Try now, please.
Try now. Yes. Share.
Okay, that's perfect.
So it works. So
It works.
I will do stop share now.
Yes, and
then you can
your
put
slide back.
Yeah. And I
That's
will also mute myself. And then
Okay.
when you need me,
I'll
need me to become active, you tell me, and then I'll.
Yes, I will.
But at least you know I'm here.
Yes. So back in a minute. Bye now. So, good afternoon, welcome everybody. This is another Euro-NMD webinar, and today we have the pleasure of welcoming Annemieke Hartsma-Roos to deliver thoughts about the basics of genetic approaches for neuromuscular disorders using DMD, so Duchenne muscular dystrophy, as a paradigm. Annemieke is a professor of translational genetics at the Department of Human Genetics at the Leiden University, and she has an interest in development of antisense mediated exon skipping therapy for Duchenne muscular dystrophy. That was the theme of her PhD thesis as well. She has a visiting professorship at the John Walton Muscular Dystrophy Research Center at Newcastle University. And in 2014, Annemieke was accepted in the Young Academy, the junior section of the Dutch Royal Academy of Sciences, consisting of the 50 most prominent scientists under 45. In 2009, she received the prestigious VD grant, and in 2011, she received the Duchenne Award for her work and dedication in the Duchenne field. And since 2016, every year since then, she was elected as the most influential scientist in the Duchenne field, based on publications of the past 10 years by Expertscape. And she's currently developing she's currently the chair of the TreatNMD Alliance as well. So welcome, Annemieke. Thank you very much for agreeing to deliver this webinar, and I will pass on the floor to you, stopping my sharing. So don't forget to share your screen, please, and unmute yourself.
Thank you, Antonio. So I think you now should all be able to see my screen.
Yes, we are.
Yeah, and it's even in full screen. So thank you very much for the opportunity to present here on the basics of developing therapies for Duchenne muscular dystrophy. And the focus will be on the genetic therapies. Here are my disclosures. I'll give you the executive summary. So as a co-inventor of some patents on exon skipping, I'm entitled to a share of royalties. I'm also consulting. I'm member of a scientific advisory board for several companies. And that money all goes to LUMC, the reimbursements. And I also have presented during symposia organized by companies. And also there, the honoraria go to the LUMC. So what we'll discuss today are the genetic therapies for Duchenne muscular dystrophy. And I will start with a brief introduction about Duchenne and dystrophin and the genetics involved. And then for the different genetic therapies, gene addition, stop codon read-through, exon skipping, and genome editing, I'll explain how they work, what the current state of the art is, and also some challenges and considerations towards the future. So we'll start with Duchenne, and I assume that you're all familiar with Duchenne. And Duchenne is characterized by progressive muscle wasting and really by a progressive loss of motor function. You can see on the top left, you see a very young patient. You don't really see that much, but from an early age, from around 1 to patients already show symptoms. They have difficulties getting up from the floor. They frequently fall. You can see the lordosis position in the 2 standing boys. Most patients lose ambulation around the age of 10, 12. Then also upper arm function declines. And eventually patient needs assisted ventilation, usually around the age of 20, and prematurely die in the 2nd to 4th decade of life. So it's a disease characterized by continuous loss of function. And the disease is caused by problems in the dystrophin protein. And dystrophin acts as a shock absorber in skeletal muscle fibers by connecting the cytoskeletal actin to the extracellular matrix. And what's interesting in the dystrophin protein is that the functional domains are located at the beginning and the end of the protein. So at the beginning is the domain, the actin binding domain that binds to the cytoskeleton, and at the end is the dystroglycan binding domain that binds to the extracellular matrix. And by this connection, dystrophin provides stability to skeletal muscle fibers during contraction. Now, what happens in Duchenne patients is that most patients have a deletion of one or more exons. This disrupts the reading frame, protein translation is truncated prematurely, and its linker function is lost. And you can ask what would happen if the frame would be maintained. Well, this is also found, but this is associated with a different disease called Becker muscular dystrophy. So if the reading frame is not disrupted, as is here shown on this example, exon 48 to 51 are deleted, but the reading frame is not disrupted, so protein translation can continue. And because in dystrophin, the important domains are located at the beginning and the end, having an internal deletion results in a partially functional protein and a less severe disease. So Becker patients generally experience a later onset and a slower disease progression compared to Duchenne. So if we compare these diseases and we use an example, one example that I like to use to explain this to parents is the carabiner. So this you use when you go mountain climbing, of course, and then you have these two functional domains that click to your harness and click to your rope, and you have an elastic piece in the middle. Now, what happens in Duchenne patients is that only the beginning of the protein can be made, so you can't make the connection to the rope. And in effect, this doesn't impair your function, your ability to climb the mountain, but you can afford yourself any mistakes. And that's what happened in Duchenne patients. Normally, you have the shock absorber, but without the shock absorber, any mistake will cause damage. Like it would if you make a mistake while climbing the rocks. Now, in Becker patients, this elastic piece is slightly shorter, but the two functional domains are there. So if you fall, the fall may be less comfortable than with the normal carabiner, but you won't crash. So what happens is there's no dystrophin, you will get muscle damage, as I just explained. And this will be repaired, but there will also be inflammation. And normally, if there's muscle damage in a healthy individual, these processes are sequential. So you have muscle damage, you have the inflammatory response to clear away the debris and the necrosis, and then you will have repair. Now, in Duchenne patients, there's chronic damage. So there's chronic inflammation, and this will inhibit the repair. And normally this is good. You don't want to repair until you've cleared away the damage. But now, because there's chronic inflammation, the repair is continuously inhibited. And even the places where the damage has been removed, the debris has been removed, the repair is inhibited. The chronic inflammation will also lead to oxidative stress. It will lead to fibrosis, and the oxidative stress in turn will also lead to fibrosis. And once there is fibrosis, this will inhibit repair. There's also less blood flow through the muscle because the NMDs enzyme is not located where it should be. So there's less vasodilation, and this leads to more muscle damage and also oxidative stress. And finally, there's too much calcium ions in the muscles. The mechanism is not really known. The stretch-activated calcium channels may be more active, maybe due to the damage. It may be a combination of factors, but it's clear that there is too much calcium ions within the muscle fibers. This leads to damaged mitochondria, which in turn leads to oxidative stress. And all these processes help steer away from muscle repair and towards replacement by fibrotic tissue, by adipose tissue, loss of muscle tissue. And this is then also accompanied by loss of muscle function. So because all these pathways that I showed, they all are caused by the fact that no dystrophin is there. And there's many therapeutic approaches that try to address one of these pathways. However, all of them are downstream of the lack of dystrophin. So an obvious therapeutic choice is to replace the missing dystrophin. And those are the therapies that I'll discuss today. So there is gene addition, stop codon read-through, exon skipping, and genome editing. And I'll go through them one by one, starting with gene addition. So the idea of gene addition is that because a gene is not functional, we will provide the cells with a functional copy of the gene. and in this case, the cDNA. The DMD gene is huge. It's 2.2 megabases. There's no viral vector that can contain that. So what we do, we use the cDNA. So only the exons, only the coding region we provide the cells with. We use viral vectors to deliver these genes. And of course, because Duchenne is a disease that primarily involves the skeletal muscle, we need a viral vector that goes to muscle. Well, there's only one that really does this quite efficiently, and that one is called AAV, adeno-associated virus. The problem with AAV is that it's small. The reason that it can go to muscle is that it's small, but because it's small, also the capacity is very limited. So only 4.5 kilobase DNA can be inserted. Well, the cDNA, so all the exons of the dystrophin gene are 11 kb, so it doesn't fit. So what the field had to do was to generate a microdystrophin, a dystrophin that is very small, contains only the crucial domains with a muscle-specific promoter that is small enough to fit within this AAV. And they have been successful. So there's 3 examples here. So at the bottom, you see the full-length dystrophin protein. So these are all the different domains. And again, this is an important domain. And the domain at the end is important. And then you can see on the top, the 3 different microdystrophins that are now tested in clinical trials. And it's drawn to scale. So you can see really that these microdystrophins are really very, very small. They're much smaller than the smallest Becker dystrophin that we observe. What do they contain? They contain the actin binding domain at the beginning. They contain the cysteine-rich domain that binds to the extracellular matrix. And then for the different microdystrophins, they either contain 4 or 5 of the central rod domain. They contain 2 or 3 hinge regions. And that is how they differ. But in the end, what they have are the 2 crucial domains that bind to the actin cytoskeleton and the extracellular matrix. And like I said, these 3 microdystrophins are currently tested in clinical trials. But before that happened, a lot of work had to be done. So these microdystrophins have been tested in mouse and dog models for the disease, and this resulted in therapeutic effects. There were also some challenges that had to be overcome. And I'm now in 3 sentences, I'm summarizing up to 10 years of work. So the first thing that had to be solved was the upscaling. And gene therapy, so AAV-mediated gene therapy for eye diseases has been approved. But what you need to treat muscle is 250,000 times more. And just think, if you make 1 liter of soup, or if you may have to make 250,000 liters of soup, there's a big difference, not just in volume, but also in the tools that you need to produce it and the systems that you need. So that took a lot of time, but large-scale production is now feasible. Body-wide delivery is also feasible. So at the beginning, people try to deliver maybe to all the muscles in 1 arm or 1 leg. Now the system has been improved further, so body-wide delivery is feasible. And of course, if you inject a virus, there will be an immune response. So it's clear that you need to suppress the immune response. Currently, this is done by corticosteroids. And also you need to pre-screen the patients, because the viral vector that you use, if the patient has already seen this virus before, then they are precluded from treatment because of the risk of a massive immune response. So where are we at? So I said already, there's 3 companies that do these gene therapy trials. They're Sarepta, Solid, and Pfizer. And I will go through them. So what we know is that Sarepta has treated at least 4 patients, but a new trial is ongoing, and we don't really know how many patients have been treated in the trial. Solid has treated 6 patients and Pfizer has treated 13 patients. If you look at the age, you will see that the Sarepta patients are younger than the patients involved in the Solid and the Pfizer trial. And if you look at the dose, you will see that they vary a little bit. First of all, they're massive. So 2 times 10 to the 14th viral particles per kilogram really is a lot of virus. Solid tried also a lower dose. Pfizer has tried a slightly lower dose and a slightly higher dose. But of course, what's more important is did this result in any dystrophin production? And if I say dystrophin here, I mean microdystrophin. So the Sarepta trial observed that about 80% of the muscle fibers that in a biopsy that they took started to make microdystrophin. They also looked at Western blotting to quantify it, and they saw that in total there was 74% of dystrophin compared to normal. If we look at the solid data, so they used first a low dose. With the low dose, about 10% of the fibers produced about 5% of normal dystrophin. When they used a higher dose, they saw 50% of positive fibers producing around 10 to 15% of dystrophin. And of course, so bear this in mind, if a fiber is positive, that doesn't mean that it produces 100%. It can also produce lower than normal amounts. So the Pfizer study also used two different doses. For the lower dose, they saw about 28% positive fibers at total levels of 24. And at the higher dose, they saw close to 50% of positive fibers and close to 50% dystrophin levels. Unfortunately, Pfizer used a mass spec analysis to quantify their dystrophin levels, while Sarepta and Solid used a Western blot. So we cannot directly compare the levels of Pfizer with Sarepta and Solid. So we don't know whether it's more or less. In all trials, side effects were observed. In the Sarepta trial, there have been no serious side effects yet, but there has been nausea and there have been elevated liver enzymes observed in treated patients. The trial has been on hold once, but this was due to a manufacturing problem that has been resolved, and the trials have been initiated for Sarepta. The Solid trial experienced two severe adverse events where patients had to be hospitalized. This was due to very severe inflammatory responses. And after the first adverse event, the trial was put on hold. They made a new managing protocol, and then there was a new adverse event, and the trial has then been put on hold again, and it's still on hold. At least last week when I checked, it was still on hold. Pfizer has seen 3 serious adverse events and also patients that had to be hospitalized. Again, serious inflammatory responses, and 2 patients had transient renal failure, needed dialysis. Fortunately, all these patients have recovered and are doing okay now. The Pfizer trial has not been put on hold. So what is now ongoing, what is planned? So Pfizer has completed their first trial and is now preparing for a phase 3 placebo-controlled trial in 99 patients using a dose of 2 × 10⁻⁴ viral particles per kilogram, which is less than the high dose that I showed on the previous slides, but apparently they have a new manufacturing protocol, and this is equivalent to their previous high dose. This is what they claimed during a presentation. So they're planning the trial, and 1 in 3 patients will receive placebo. It's double blind, but the patients that receive placebo after a year will receive the gene therapy if they've not developed antibodies against AAV by then. Sarepta has a placebo-controlled trial currently ongoing in 41 patients, again, young patients, and this is fully enrolled. They have also received fast track designation from the FDA, and they're planning a trial in older patients, but this is very preliminary. So there's not any data known yet, but they have announced that they want to also test this in older patients. And like I said, the Solid trial is still on hold. So there's several outstanding questions and considerations for the gene therapy. The first thing is the manufacturing. So it's now possible to manufacture for larger groups of patients, but if this would be approved, then this would mean that this is not a mutation-specific therapy. So in theory, everyone who doesn't have an antibody response against AAV would be eligible for treatment. So that would mean that the manufacturing needs to increase a lot. And it's viral production is challenging, so that will always be a challenge. But it's much better than it was 5 years ago. Unfortunately, not all patients will be able or will be eligible for this therapy, because if they have already an existing immune response against the serotype, the AAV serotype used for the delivery, they cannot be treated because this will induce such a massive immune response that is likely a problem. Of course, research is ongoing to try and see how to counteract that, maybe plasmapheresis, maybe other ways to get rid of that immunity. For now, patients who have an immune response against the AAV serotype are excluded from treatment. And this also means that retreatment is not possible, because even with the immune suppression, the patients will respond to these immense doses of viruses. Well, what we don't really know is the functionality of the microdystrophin. So you know that it's really small, it's much smaller than the smallest Becker protein. So we have no evidence that this will really be functional. There are some anecdotal stories where the patients that were treated in the gene therapy trials do better, and so their walking distance in the 6-minute walk test seemed to improve, and their North Star ambulatory assessment skills seem to improve. However, first of all, most of these patients are very young, and we know that these skills will go up anyway in young patients. And secondly, they're treated with huge doses of steroids, which we also know can improve these tests. So we really have to wait and see whether there's a functional effect of the microdystrophin. And so far it's open label treatment only, so we cannot draw firm conclusions. Then the question is longevity. Now, normally you would think if we do gene therapy, that's one-time treatment and you're done for life. Now, the problem here is that we know that the microdystrophin is not going to be fully functional. At best, it will be partially functional. We know that Becker patients also have muscle turnover. So we know that even the muscles that have this microdystrophin, with time, there will be turnover of muscle. Now, AAV doesn't integrate in the DNA. The AAV genome stays within the cytoplasm of the muscle fibers. And that means when there's muscle fiber turnover, these AAV transgenes, these microdystrophin transgenes, will get lost. Now the question is, how long will this take? Will this take 1 year? Will this take 5 years? Will this take 10 years? And that will, if it's 1 year, then it's going to be very difficult to treat patients every year with this, because again, there's the immune response. If it's 10 or 20 years, it's a different story. This is something that we do not know yet. And then one thing to consider is the long-term and the short-term safety. And it's known now that injecting these huge amounts of viruses is not without risks. So then we continue with the second genetic therapy, and that is stop codon read-through. And while the gene addition is mutation A-specific, stop codon read-through only applies to patients that carry stop codon mutations. So these are mutations where the code for one amino acid is changed into the code for a stop. So you have here at the beginning of the transcript, you have the start signal. Normally at the end, there's a stop signal to indicate that translation can stop. If one amino acid a code is changed into a stop code, this will be used. The reading frame here is not disrupted. The frame is maintained. And that means that if you could suppress this premature stop codon, you would be able to make a normal dystrophin. Now, there's several compounds that can do this. And one that has been in clinical development is called ataluren or Translarna. So, like I said, it's a stop codon read-through drug. It's an orally delivered compound. You have to take it 3 times per day. It was tested in healthy volunteers. It was safe. And it's been tested in 2 clinical trials in Duchenne patients. And it was first tested in a phase 2b trial. And there, the primary endpoint was the 6-minute walk distance. So they looked at the baseline, how far patients were able to walk, and they looked after 1 year of treatment. And the endpoint was not met. So there should have been a difference of 30 meters between treated and untreated or placebo treated, and it was less than that. Now, they did post hoc analysis to look at whether maybe everyone that was included should have been included. So maybe there were patients that had just had a bad fall or were recovering from severe flu, things like that. So they excluded all the patients where they thought, well, maybe we shouldn't have included them. And then, lo and behold, they did find a difference of close to 50 meters between treated and untreated. Now, of course, this is post hoc analysis, so it's less convincing, but still they went to the EMA with this data and they got conditional approval. So the EMA said, well, you have a very good safety profile and we are convinced that this seems to slow down disease progression. However, you need to collect more data. So that is where the second trial came in, so the phase 3 trial. So on the left here you see studies 007, that's the study that I just showed. So the post hoc analysis is shown here. And that was focusing on patients that were able to walk at the start of the treatment between 300 and 400 meters. And the reason for choosing these patients is that in this patient group, if you follow them for a year, you know that normally they will decrease to a certain extent, which will make it able to, within a year time, see whether they decrease less. If patients are stable, you won't be able to measure that there's less disease progression. If patients get better, it's difficult to measure whether there's a slower disease progression. So they said this specific spot, this is what we want. Now, for their phase 3 trial called DMD, what they wanted was to have more than half of the patients that fell into this criteria. So that was able to walk between 300 and 400 meters. So that was their goal. However, they did not succeed in this. So if you see, they managed to get 100 patients that were in this, this specific region. Those, again, they saw that there was a treatment effect. They walked further than the untreated patients. But if you look at all the patients combined, so also including patients that were doing less and worse, then you see that the difference was much less. So what happened? They went to the EMA again, and the EMA said, well, the problem here is that they couldn't conclude that it worked. So the data didn't confirm that it worked, but they also could not conclude that it didn't work. So what they did, they requested a third or a second confirmatory trial. So what's happening now? The drug is conditionally approved in the EU. It used to be approved for patients 5 years and older. Now it's approved for patients 2 years and older due to additional data. Every year, the company has to go through the EMA to reevaluate whether it's still effective or not, and whether it's still safe or not. It's marketed in several European countries, but it's not available everywhere. And outside of Europe, or at least in the US, it's not approved. And there's currently new trials ongoing. So this placebo-controlled trial, the second confirmatory trial, is ongoing. There's also an open label study ongoing in very young patients. And of course, the idea is that the earlier you start treatment, the more patients will benefit from this. There's also a trial ongoing that includes a biopsy. So you may wonder, well, why didn't they do a biopsy in these trials? Well, at first that was not suggested by the ethical committees, so they didn't include it. But now, of course, they want, or they were requested by the regulatory agencies to also show that dystrophin is indeed restored like it is in the cell and animal models that they've tested this on. And because it's now approved in several countries, post-marketing studies are ongoing as well. So something to consider, this applies only to nonsense mutations. So you see a nonsense mutation at the top. This is a nonsense mutation exon 35. And like I said earlier, if there is a nonsense mutation, if an amino acid chain code is changed into a stop, the reading frame is not disrupted. However, if you have a small mutation that disrupts the reading frame, so an insertion or a deletion, then the wrong frame is used. So even if you would use this for these patients, you can see there's a lot of stops here because the wrong frame is used. Etalurin is not going to do anything. It's not going to achieve anything, because even if you would manage to suppress all these stops, which you can wonder whether you will succeed, the wrong amino acids are built in because it's in the wrong frame. So regardless, the protein will not be functional. So bear this in mind, etalurin only applies to nonsense mutations and not to other small mutations and not to deletions and duplications. So then we continue with exon skipping. And exon skipping tries to modulate splicing. And a brief recap of splicing. So we have the gene, the genetic code is dispersed over the exons before the protein can be made. A temporary RNA copy is made containing both the exons and the non-coding introns. During splicing, the introns are removed. The messenger RNA is produced containing only the coding information, and that is then translated into protein. Now, we've already seen that Duchenne patients have out-of-frame mutations, and the basis of exon skipping is that we try to reframe the code. So you see here, exon 47 doesn't fit to exon 51. It would, however, fit to exon 52. So during the splicing process, what we want is that exon 51 is not recognized, that it's skipped. Well, we can do that with AONs, antisense oligonucleotides, that can bind specifically to an exon, hide it from the splicing machinery. The splicing machinery will go look for the next available exon, which is 52 in this instance. The reading frame will be restored, and our Duchenne patient can produce a Becker-like protein. So like ataluren, this is a mutation-specific approach, but unlike ataluren, this does not apply to all the different mutations. You have to customize it for different mutations. So depending on where the mutation is located, you have to skip a specific exon. Now, if the mutations are located here in the actin binding domains or at the end in the dystro-glycan binding domains, it doesn't matter whether the frame is disrupted or not. This will not produce functional proteins because these domains are crucial. Now, fortunately, most patients have a deletion in a hotspot located between exon 45 and 55. So skipping certain exons applies to larger groups of patients. And you can see here the example. So number one, exon 51 would apply to 14% of patients, 45 would apply to 9%, and so on and so forth. And understandably, the clinical development started with those that apply to larger groups of patients, because if you need to test this in clinical trials, you need larger groups, and they are only available if the applicability is relatively large. So when you want to use antisense oligonucleotides, they're basically DNA or RNA homologues. You have to modify them to increase the stability, to make them bind better to their target transcript, to make them give them improved uptake by cells, and to prevent clearance by the kidney. And there's many modifications available. The ones that are used in clinical development for Duchenne are 2-O-methyl phosphorothioate and the phosphorodiamidate morpholino oligomer. And I will call them usually the 2-O-methyl and the PMO. So there's something to consider when you deliver the AON systemically, and that is that they are very small. So normally they would be filtered out by the kidney. Now, if an AON has a phosphorothioate backbone, this will allow low and reversible binding to serum proteins, and that will prevent renal clearance, and that will therefore improve bioavailability. If an AON doesn't have a phosphorothioate backbone, that means that it will be filtered out by the kidney very efficiently and very quickly. So you will see that in some of the AON trials, they use much higher doses than others. Well, that is because of this problem. So if you have a phosphorothioate backbone, the bioavailability is higher, so you can use lower doses. If the AON doesn't have the phosphorothioate backbone, the filtration by the kidney goes incredibly fast, and you need to increase the dose in order to get something to the skeletal muscle. So for exon 51, a lot has been done. So we start with the top one, that is drisapersen, developed by Prosenza, GSK, and BioMarin. The dose that was used mostly was the 6 milligram per kilogram dose. You will see this one has a phosphorothioate backbone, so the dose is relatively low. A phase 1 trial was done, dystrophin was restored. A phase 2 trial was done. Again, dystrophin was restored. There seemed to be functional effects. A phase 3 trial was done. No functional effects were seen. And this drug was not approved by the FDA. And the main reason is that even though it seemed that there might have been a functional effect in a subset of patients, it was clear that there was side effects in all patients, including injection site reactions, but also thrombocytopenia in some patients, proteinuria in some patients. So the risk benefit ratio was not very good for this. So the FDA did not approve this, and the EMA also did not approve this drug. So then we have eteplirsen, which is developed by Sarepta. So that is a PMO. You can see it doesn't have a phosphorothioate backbone. It has a much higher dose that's used, so 50 milligram per kilogram. And the approval was based on only a phase 1 trial showing only dystrophin restoration. So a confirmatory phase 3 trial is currently ongoing to see whether there's also functional effects. But this drug has only been approved on the ability of this compound to restore low amounts of dystrophin in the order of 1%. There's currently also a trial ongoing with this compound, SRP-5051, which basically is an eteplirsen with a peptide conjugate. And this peptide conjugate is there to improve the delivery of the compound by skeletal muscle and heart. The dose has not been disclosed. That's currently used in clinical trials. So it's a phase 1 safety study. And the main question here is whether this will be safe, because many of these peptides that are used to improve uptake turn out not to be safe. So hopefully this one will be effective before there are safety concerns. But this trial is ongoing, so we don't know that data yet. Then there was a compound developed by Wave called suvodirsen. This one has a phosphorothioate backbone. You see the dose is lower. They've done a phase 1 trial, but unfortunately after treatment, there was no dystrophin restoration observed. So this development has been stopped. And finally, BioMarin is planning to work on a better exon 51 compound with the phosphorothioate backbone. But this is in a very early planning stage, so no information is available yet. So there's also 2 compounds for exon 53 skipping. Both of them are PMO, so both of them don't have a phosphorothioate backbone. You can see the dose, 50 milligram per kilogram and 80 milligram per kilogram. These compounds have been approved, but like eteplirsen, they're approved because of their ability to restore dystrophin in skeletal muscles. Functional effects still have to be confirmed. And golodirsen has been approved by the FDA, and viltolarsen has been approved by the FDA and also by the Japanese Ministry of Labor and Welfare and Health. So then there is also an exon 45 skipping compound, casimersen, developed by Sarepta. This is a PMO, so again, no phosphorothioate backbone, high doses. A phase 1 trial has been done. Low amounts of dystrophin restoration has been confirmed. A phase 3 confirmatory study to see whether there's also functional effects is ongoing. And Sarepta has submitted an NDA, so a new drug application, to FDA. And next year in February, FDA will give the verdict on whether or not this drug will be approved. In Japan, there's also an exon 45 skipping AON with the phosphorothioate backbone that's in clinical development. It currently only has a number, DS5141B. It's been tested in doses up to 6 milligram per kilogram. And in the phase 1 trial, they saw that after 12 weeks of treatment with 2 or 6 milligram per kilogram, there was exon 45 skipping in all the patients, but they couldn't see dystrophin restoration. And currently they're treating all the patients for 1 year, and they will reevaluate then. And this compound has not yet been approved. So currently there's 3 AONs approved, 1 for 51 and 2 for 53. This is only based on dystrophin restoration at low levels. Functional effects still have to be confirmed, and clinical trials to do this are currently ongoing. And in addition, work is ongoing to improve the chemistry and to improve the delivery of these compounds. But there the problem is that with the new chemistry, like the one that Wave developed, with the new chemistry, the safety profile is often unknown. So this is more challenging than going with compounds where already there's 10, 20, 30, 40 years of safety of data in humans available. Some considerations for exon skipping: it's a mutation-specific approach, so it's only applicable to subgroups. It's not yet approved in Europe, and you need repeated treatment. So there's AON turnover, there's transcript turnover, there's dystrophin turnover. So the currently approved AONs have to be infused every week with intravenous infusion. There's also work ongoing on delivering an antisense gene, and that's in development by Audentes with Astellas. And here they've hijacked a gene called U7 as an RNA that normally has also an antisense part that binds to RNA. And here they have replaced that normal part with an exon 2 targeting part, or an exon 51 targeting part, or an exon 53 targeting part. And the idea is that if you deliver this with AAV, you have permanent exon skipping, where you have a much larger dystrophin than you would with the microdystrophin. The downside, of course, is that this now also becomes mutation specific. Well, if you deliver a microdystrophin, it's not mutation specific. This work is currently ongoing for the exon 2 skipping gene therapy. A biopsy was obtained from one of the patients, but analysis is still pending. Now then finally, genome editing. And genome editing works with the CRISPR-Cas9 system. And what the CRISPR-Cas9 system basically is a GPS-guided DNA scissor. So the CRISPR, the guide RNAs, tell the Cas9, the DNA scissor, where to cut. Now, why would you want to cut the DNA? You want to cut the DNA because that will activate the DNA damage repair system, and using that you can edit the genome. So there's two ways this can be done. So on the left you can see the homologous recombination way. This is error-free. So what you use a template, and you can use this to correct small mutation. Now we can forget about the left side because this only works in dividing cells, and skeletal muscle is post-mitotic. So most likely, so far, this doesn't seem to be working well in skeletal muscle. So we need to use the thing on the right, which is called non-homologous end joining. And what this does is it's basically a glue system. So there's a cut, and the system tries to, as quick as it can, to join the two ends. During this process, small mutations will be introduced, and you can use that to correct the reading frame for Duchenne. And what this does, so it does basically the same thing as exon skipping, only now on a DNA level. So you don't have to every week give these AONs, you have to treat only once. Other advantages is that you can use it to remove multiple exons, and you can also use it to repair duplications. So we have here an example. So we have an exon 50 duplication, and at the top, what would happen if you would do exon skipping? You would have an AON targeting exon 50. And the problem here is that there's two exon 50s, they're identical. The AON will recognize both. And of course, if you skip just the first or just the second, you will restore normal dystrophin. However, since they're identical, what will happen most likely is that both of them will be skipped, and you go from out of frame to out of frame. Now, with genome editing, what you can do, you can make a scissor and a guide RNA that targets here, just before exon 50. That will then also target here, because this is the same sequence. It will cut out exon 50, and you end up with a normal transcript or normal gene. Now, for other mutations like deletions, you cannot end up with a normal transcript. With deletions, you need to cut out, you need two scissors to cut out an exon and then restore the reading frame like that. Now, there's a big hype about genome editing. So it's in the news a lot, and there's also a big hype in the Duchenne field. Why is that? Well, that is because the technique offers a lot of potential. You can do targeted DNA modification. This is what researchers were hoping for years. You can also use it to generate model systems much easier than we could. And of course, you can exploit it as a therapy. However, there are some buts still there. For DMD, there's many examples that this works. The thing is that it so far only works in model systems. So a couple of years ago, there were 3 back-to-back papers in Science showing that genome editing worked in the MDX mouse model. Now I'll just show you how that works. So the MDX mouse model has a stop mutation exon 23. Because exon 23 is in frame, if you use 2 scissors, you can cut at both ends. The exon will be removed, and the non-homologous end joining system will just fuse the 2 ends. You get rid of exon 23, and bypass the mutation without disrupting the reading frame. Now, they tried this in mouse and they did local delivery and then looked at DNA. So you can see that on the top here, you can see the smaller band. This is where exon 23 was deleted. And if you look at bar graph C, this looks quite promising, but look at the scale. This is 2%. So in 2% of the nuclei, they were able to delete exon 23. They also showed it on RNA level. They showed on protein. So here you can see protein restoration and also with immunohistochemistry. So what you see is it works, but it's not that efficient yet. So what has been achieved, like I just said, the exon 23 has been deleted and and dystrophin has been restored at a limited efficiency. If you want to do this systemically, you will need a viral vector system. We're back to AAV. And because Cas9 is a big protein, you need just one AAV to deliver Cas9 and then the second AAV to deliver the guide RNA. So that means it's a two-component system. Now, what some people say, some people use the phrase CRISPR cure. And for Duchenne, unfortunately, that won't be the case, because what we will do, we do exon skipping at DNA level. So the proteins that are produced will be partially functional and not fully functional. And of course, also with all these therapies, the therapeutic effect depends on the time of intervention. If you intervene at a specific time, unfortunately, damage that has been accumulated won't be restored. So you will slow down disease progression at the time of intervention, which in my mind doesn't mean that it's not a cure. Still, there is a big hype surrounding genome editing. And I show you here a recent example. So there was a publication where they did genome editing in a dog model. And for 4 dogs, they did local intramuscular treatment and they saw local dystrophin restoration. They also tried systemic treatment with a low dose, not successful, and with a very high dose in 1 dog. And what they showed in that dog was that there was dystrophin restoration, which was nice, but they didn't show anything else. So there were no functional studies done. They looked at 1 time point, so they only know that there's short-term restoration of dystrophin. They didn't look at side effects, etc. So it was a very small study, and credit to the authors, they didn't hype it. However, the story was picked up so there was also an editorial in Science. So this was published in Science. There was an editorial in Science that hyped it a little bit more, and then it was picked up by the media. So the first one is quite modest. So CRISPR fixes a muscular dystrophy. Well, I wouldn't say it fixes because there's a partially functional dystrophin. They didn't look at any function. And then it says treatment repairs the gene in beagles by further mutating it, but trials are far off. So that's okay. Well, 2 hours later, CRISPR halted muscular dystrophy in dogs. Are humans next? Well, already a little bit more excessive. And then a couple of hours later, this appeared. CRISPR genome editing successfully stopped Duchenne muscular dystrophy in dogs, and human trials are next. And you can see how quickly this was picked up and how quickly it exacerbated into something that is completely unrealistic, because the disease wasn't stopped in these dogs. They didn't even look at that. They only showed that they could restore a partially functional dystrophin. So there's other outstanding issues, and that is first, how do you deliver these genes? Because you need AAV, you need a 2-component system, and even if you manage to deliver it, it's not like microdystrophin, where if the transgene is expressed, you're done. No, you need to then have the Cas9 do its cut. You have to have the repair system repair it. So it's less efficient than the microdystrophin. And then of course there's also a safety aspect. So how specific are these Cas9s? And we know now that they also cut where they shouldn't cut. So that is something that definitely needs to be addressed. So in summary, there are a couple of genetic therapies that are approved, 1 in Europe, 3 in the US, and 1 in Japan. There are several more in clinical trials, and there's also those that have been approved. There's confirmatory studies ongoing in clinical trials, and the genome editing currently is only in preclinical trials. And then I will leave you with this last slide, which is of the Duchenne Center Netherlands, just to show that in the Netherlands, everyone who works on Duchenne in different cities and different institutes collaborate, and we also collaborate with the patient organizations. So with that, I will end, and thank you all very much for your attention.
Thank you, Annemieke. You can stop your sharing if you want, and yep. Yes, and like that we'll see you much better. So this was a very exciting exposition, and you are a very good speaker and make things that are very complex quite easy. And although at the moment we just have one question from Arjit Bolmik on, but I don't know if Arjit wants to talk. He can unmute himself now. Everybody can talk now. So he's asking about the administration route. So if all these drugs are intramuscular. So I don't know if you want to comment on those particularities.
Yeah. So hi, Annemieke. Sorry, first of all, that's a great talk. Thanks. My name is Arjit and I work for Regeneron. I'm just curious if you have any update on any targeted muscle delivery approaches for ASOs so far yet in DMD or any other diseases. Thanks.
Yeah. So I'll first address your first question, and then I'll go into your second question. So it probably became clear during the presentation that most of these therapies are delivered systemically. For the gene therapy, they do intravenous infusion of the viral particles. For exon skipping, they do intravenous infusion of the antisense oligonucleotides. For the etelurin, it's orally delivered, but of course with the aim that it's systemically delivered. And eventually with the genome editing, they also want to do it systemically. Now for the targeted delivery, there's a lot of work ongoing on that, because skeletal muscle and also heart are tissues that are notoriously difficult to treat and to deliver things to. So there's work ongoing where they try to link muscle-specific antibodies, so antibodies that recognize receptors on skeletal muscle, in hoping that will improve delivery. And I know Avidity has some preclinical work where they coupled oligos to a transferrin receptor, and that seems to improve uptake and exon skipping levels in the mouse. So that is ongoing, and I know that there's several other people that also try with nanoparticles, try to improve delivery. But the problem really is that skeletal muscle is difficult to deliver to because the endothelium is quite dense. So you need small particles, and also many of these targeted approaches, they use proteins or receptors that are expressed on skeletal muscle, but a lot of these are also expressed on liver. And liver is generally much better able at taking up things than skeletal muscle. So what we've seen, so we've tried things also with muscle homing peptides, and what we generally observe, yes, we see more uptake in skeletal muscle, but even more improvement in liver. So there's a lot of work ongoing, but there's no magic bullet yet.
Great. Thanks a lot.
Okay, I don't see any more questions. Yes? Okay, so if no one has a question for the moment, I have one. So I think that dystrophin is an obvious biomarker for this condition. I would compare it to insulin in the case of diabetes, probably. But what I do not know is how far have the functional validation of the dystrophin changes have convinced you, not only in terms of preclinical and other evidence like that, but during the clinical trials, how do you think that dystrophin has performed as a biomarker?
Yeah, so I think that the thing is that the views on how dystrophin can be used as a biomarker differ between the European regulators and the American regulators. So I think dystrophin is a biomarker for any therapy that aims to restore dystrophin. Of course, dystrophin is a biomarker. You expect it to increase after the treatment. So in that respect, it's a biomarker. The challenge is that it's very difficult to properly quantify because it's a low abundant protein. It's very long, so it's difficult to really quantify it properly, especially if you have really low doses. And I think that's, or low levels that are produced. And I think that's, so with the microdystrophin trials, what you see are levels like 20, 30, 40, 50, 80% dystrophin. Well, I'm quite convinced. I mean, if it's not 80%, at least there's 70%. I'm quite convinced that there are good amounts of dystrophin. Now, with the exon skipping therapies, the levels of dystrophin are much lower. So we're talking about 0.5% increase, 1% increase, 5% increase. And that is much more difficult because if it's 0.5%, it's really difficult. So is this really an increase or is this just natural variation? And another thing is that if it is an increase, is 0.5%, so 0.5% dystrophin, is that going to be enough to slow down disease progression? So I always say some dystrophin is better than none, but of course there are limits. And I think also what is clear, I mean, there's dystrophin restoration, so the drug is doing what it's supposed to be doing, but what these confirmatory studies will need to show is whether these low levels are sufficient to slow down the disease progression. And of course, once this has been confirmed, then we know that measuring dystrophin is, then it becomes a much better biomarker. Then it becomes not just a biomarker to show that the approach works, but also to show that likely at these levels that you now measure, there will be a functional effect in 1 year, 2 years, 3 years.
Excellent, thank you. Francina Munel, do you want to voice your question or shall I read it? Francina? Well, in that case, I'll read it. So Francina Munel is asking, do you know how many patients were excluded from the gene therapy clinical trials due to neutralizing antibodies against AAV9?
So I don't know. I know that depending on where you live, there's 20% up to 50%, up to even higher percentage of people that have these neutralizing antibodies. I also know that the older you are, the higher the chance that at some point you've encountered this virus. But I wouldn't know the exact numbers. And I think so, if you think, for instance, of the Pfizer trial, the placebo-controlled trial, where they now want 1 in 3 will receive placebo, and then after a year will be treated with the virus. I mean, if that patient sees the virus within that year, that would of course be awful, because then they're contributing to the trial, but won't be able to receive the treatment. So those are, yeah, say more ethical concerns. I know that some patients have the neutralizing antibodies and were excluded, but I don't know the numbers, but I know that it has happened, unfortunately. I mean, it's not unfortunately, I mean, they make the right call to exclude these patients because there's too much risk involved. But of course, it's sad that they have to exclude them.
So what would you say would be the ideal changes that you would like to see in order to improve future drugs for DMD? What would you put in your wish list?
So I think the gene therapy looks very promising, and there's very high levels of dystrophin, but I'm worried about the risk. So I'm worried about these severe adverse events. So it's definitely not without risk. So I think one thing there, I would want to see that there's safer viral vectors. I also worry that this microdystrophin is not functional enough. So if we had an AAV that could deliver larger dystrophins and be more safe, that would be great. But it's probably not a realistic wish. So then, no, I'm of course from the exon skipping field. So of course, my biased answer is that I hope that in the near future, more efficient delivery of exon skipping compound will be achieved. If you look at the Vilto-Larsen trial with the very high dose, so they used 80 milligram per kilogram, but they achieved about 5% dystrophin levels. Well, 5% is a lot more than 1%, and with 5%, I really do think that it's likely that there will be a slower disease progression. So I hope that will work out. But then, of course, there's the problem that this won't be a therapy for all patients. So I hope in parallel there will be both better viral vectors that can deliver to muscle in a safe and efficient manner in a larger cargo, and then at the same time also the exon skipping compounds to improve. And then that's not even, we haven't even gotten into the other compounds that improve muscle mass, muscle quality, that reduce inflammation, that reduce fibrosis. All of these hopefully also will make a difference.
Okay. So thank you very much, Annemieke. I think we don't have any at the moment. This was rather an informative talk. People will be able to review it later on the website of our network, the Euro-NMD network. And thank you very much for your collaboration, and I hope to see you very soon. Okay. Bye now.
In person.
Bye, everybody. Thank you.
Bye.
Bye-bye.